wanted to know the odds of finding any functional protein whatsoever within such a space. That number would make it possible to evaluate chance-based origin-of-life scenarios, to assess the probability that a single protein, any working protein, would have arisen by chance on the early Earth. Stephen C. Meyer, Signature in the Cell, pages 210 to 211. Fortunately, Axe's work provided this number as well. Axe knew that in nature proteins perform many specific functions. He also knew that in order to perform these functions, their amino acid chains must first fold into stable three-dimensional structures. He first performed experiments that enabled him to estimate the frequency of sequences that will produce stable folds. On the basis of his experimental results, he calculated the ratio of A, the number of 150 amino acid sequences capable of folding into stable function-ready structures, to B, the whole set of possible amino acid sequences of that length. He determined that ratio to be 1 to 10 to the 74th power. In other words, a random process producing amino acid chains of this length would stumble onto a functional protein only about once in every 10 to the 74th power attempts. In conversations with me, Axe has compared the odds of producing a functional protein sequence of modest 150 amino acid length at random to the odds of finding a single marked atom out of all the atoms in our galaxy via a blind and undirected search. Believe it or not, the odds of finding the marked atom in our galaxy are markedly better, about 1 billion times better, than those of finding a functional protein among all the sequences of corresponding length." End quote. And the problem for chance is even worse than that. That's because these calculations dealt with the possibility of stumbling onto a functional sequence. And even if you had the correct sequence or order of amino acids, for a protein to form, you would still need only left-handed amino acids in every spot of the chain. Scientists explain that every amino acid found in proteins has a left-handed and a right-handed version. Working proteins only accept left-handed amino acids. Thus, the possibility that each amino acid along the 150 amino acid chain would be left-handed is 1 chance in 10 to the 45th power. Moreover, peptide bonds would also be required to link each amino acid with the next one in the chain. In experiments with amino acids, it has been discovered that peptide bonds only form about half of the time. Thus, the chance that every amino acid in the chain would have a peptide bond is also 1 chance in 10 to the 45th power. Taking all of these facts into account, Meyer concludes, quote, "Axe's improved estimate of how rare functional proteins are within sequence space has now made it possible to calculate the probability that a 150 amino acid compound assembled by random interactions in a prebiotic soup would be a functional protein. This calculation can be made by multiplying the 3 independent probabilities by one another. The probability of incorporating only peptide bonds, 1 in 10 to the 45th power. The probability of incorporating only left-handed amino acids, 1 in 10 to the 45th power. And the probability of achieving correct amino acid sequencing using Axe's 1 in 10 to the 74th power estimate. Making that calculation, multiplying the separate probabilities by adding their exponents, gives a dramatic answer. The odds of getting even one functional protein of modest 150 amino acids by chance from a prebiotic soup is no better than 1 chance in 10 to the 164th power. It is almost impossible to convey what this number represents, but let me try. Consider that there are only 10 to the 80th power protons, neutrons, and electrons in the observable universe. Thus, if the odds of finding a functional protein by chance on the first attempt had been 1 in 10 to the 80th power, we would have said that's like finding a marked particle among all the particles in the universe, a much larger haystack. Unfortunately, the problem is much worse than that. With odds standing at 1 chance in 10 to the 164th power of finding a functional protein among the possible 150 amino acid compounds, the probability is 84 orders of magnitude, or powers of 10, smaller than the probability of finding the marked particle in the whole universe. Another way to say that is the probability of finding a functional protein by chance alone is a trillion times smaller than the odds of finding a single specified particle among all the particles in the universe. And the problem is even worse than this for at least two reasons. First, Axe's experiments calculated the odds of finding even a relatively short protein by chance alone. More typical proteins have hundreds of amino acids, and in many cases their function requires close association with other protein chains. For example, the typical RNA polymerase has over 3,000 functionally specified amino acids. The probability of producing such a protein, and many other necessary proteins by chance, would be far smaller than the odds of producing a 150 amino acid protein. Second, as discussed, a minimally complex cell would require many more proteins than just one. Taking this into account only causes the improbability of generating the necessary proteins by chance, or the genetic information to produce them, to balloon beyond comprehension. If we assume that a minimally complex cell needs at least 250 proteins of, on average, 150 amino acids, and that the probability of producing just one such protein is 1 in 10 to the 164th power, multiplied by itself 250 times, then it would be 1 in 10 to the 41,000th power. End quote, Stephen Meyer's Signature in the Cell, pages 211 to 213. Therefore, even if one assumed that the constituent parts of a small protein were somehow able to come into existence out of nothing and then survive on their own, something that's totally unscientific and unrealistic, there is absolutely no chance that a functional protein, or the many other proteins required for a minimally complex cell, would form randomly. A functional protein would also require a system of other proteins and molecular machines for it to perform its role.